All right, everybody, we're going to move on to chapter 11, which is the nervous system. So this is another chapter that's quite large and has a lot of information. So I will try to point out when there's information that's just nice to know and that you don't really need to focus on memorizing 100% of it. And this is one that I will do in sections just so it's not a super long lecture at once. That way you guys can kind of digest it in parts. Let's go ahead and get into it. So you can think of our nervous system as our electrical system. So kind of like how your house has electric wires that run all through it, that's our nerves in the body. So we use those electric signals to transmit messaging. And so our nervous system has three primary roles, which you can kind of think of as steps in order. So initially, we're going to utilize our sense organs, nerve endings to actually detect what's going on both inside and outside the body. Once we detect what's going on, we now have to process that information and we have to integrate it. So our brain quickly thinks back through past experiences, and it'll determine what the proper response to that input is. And that results in a command being sent to our muscle, gland, whatever we need to make a change based on that info that we just received and integrated. So we talk of the two major divisions of the nervous system being the central nervous system, or our brain and spinal cord, and then the peripheral nervous system, which is our kind of everything else nerve network. So anytime we're talking about the brain or the spinal cord, we're talking about the central nervous system. The remainder of our nerve system or nerve network is the peripheral nervous system. All right, you'll see this little diagram pop up throughout the PowerPoint. And so this is really just to help kind of keep you on track in terms of what our organization is. So like we just talked about, we have our two big divisions at the top here, our central nervous system and our peripheral nervous system. And we'll go through individual parts of a little more specific as we move down the chain. Okay, before we get to that, we'll talk about just our neuron structure. So that picture on the left there is a singular neuron. You have your cell body, which is also known as the soma. That's going to be your control center. You can see it contains the nucleus there. Your dendrites are these branches off of that cell body. And so they receive info and they can also conduct signals. Anytime we're sending a signal away from the cell body, it's actually going to go down this axon here, which is this longer extension. You'll notice there's these little nodes and kind of bundles of myelin, it's called. And so that whole extension is the axon, and the myelin sheath is this covering around it. And so we'll talk about the support cells of the nervous system in a little bit. But Schwann cells are what create the myelin sheath in our peripheral nervous system, and oligodendrocytes create the myelin sheath in our central nervous system. Where each little sheath or cell meets is called a node of Ranvier. And so that's important in our nerve conduction, and we'll talk about that when we get into the steps of impulse conduction in a little bit. And then at the end, we have a synaptic knob. So if you remember when we had our muscle chapter, we talked about the synapse between the motor neuron and the muscle fiber. So that's another synapse. So they call that the synaptic knob. That's where the ending of our axon is, and that's where the neurotransmitters are contained to be released to propagate the signal. Okay, so neuroglia or glial cells are the word used for the support cells of the nervous system. So we already talked about oligodendrocytes and Schwann cells in the last one. So oligodendrocytes are going to form that myelin sheath in the central nervous system, and Schwann cells are going to form your myelin sheath in the peripheral nervous system. Your ependymal cells, they line your spinal cord and the cavities of the brain. And so some of those cells will actually produce cerebrospinal fluid or CSF. And some of them will have cilia, those little extensions, to help keep that CSF moving. Microglia, that's kind of our immune cells or our white blood cells of the nervous system. And so they perform phagocytosis on unwanted visitors. And then our astrocytes form the blood-brain barrier. So they're another protective system. And the blood-brain barrier is a super, super selective and picky barrier so that things that are not supposed to get up to our brain cannot enter. And we'll talk about the astrocytes in another slide. Okay, so this is a zoomed in version. So this yellow area, that would be considered your axon. And then this wrap here is the myelin sheath formed by the Schwann cell. So remember, this would be the peripheral nervous system. And so it basically lays down a ton of layers of cell membrane to insulate the axon. And it does this because that means it can send the signal more quickly. One other thing to point out in this picture is the term neurolemma, and that's the outermost layer of that Schwann cell. And so when we have an injury to a nerve, if that neurolemma is completely severed, the chances for that nerve to regenerate and heal is much lower. So nerves are also already very, very finicky when it comes to healing, but they do much better if that neurolemma is somewhat intact. And then, so this is an example in the central nervous system. So unlike the Schwann cells, which kind of individually wrap around each section, an oligodendrocyte can kind of branch out to wrap around. And so there's no neurolemma because of the way this oligodendrocyte forms the myelin, which means if the neuron gets damaged, there's really no way for it to regenerate. So that's why damage in the central nervous system is much less reversible than damage in the peripheral nervous system. Okay, here's our astrocytes that form the blood-brain barrier that we talked about. You guys don't really need to know a ton of specifics about it. This is a YouTube video that goes a little bit more in depth with it. All you guys really need to know is that the astrocytes wrap around the capillaries to kind of be our protective system. And so its job is to not let foreign substances into the brain. And so this blood-brain barrier, on the kind of a clinical note, is a real tricky thing for pharmacists and those that develop pharmaceutical drugs to deal with, because there's certain stuff that we need to get to the brain medication-wise, but we have to make it in a way that it can get past the blood-brain barrier in our astrocytes. Okay, a few different classifications of neurons, and it mainly talks about how they communicate or what signals they deal with. So you have your sensory neurons, which are those that detect stimuli. And, for example, kind of the sensory neuron would feel the heat of the candle. And so those are also known as afferent neurons. So you can remember afferent arrives. So afferent arrives. So that's we're detecting that stimuli. On the opposite end, you have your motor neurons. And so they're going to be the ones that relay messages for actual movement. So in this case, our sensory neurons feel the heat of the candle. Our motor neurons pull us away. So they're known as efferent neurons. So efferent exits. Afferent arrives, efferent exits. Then you have your inner neurons, which are kind of in the middle. They connect. Those are only in the central nervous system. So you will not find these in the peripheral nervous system. That is important to know. And so they can receive and process information, but they kind of, like I said, they connect the sensory and motor pathways. So they do a little bit of both. All right, another way we can classify neurons is with the polarity, or in this case, it's really how many extensions it has. So the most common is our multipolar neurons. And so there's one axon, just one branch that ends in our synaptic nodes, knobs. And then we have our dendrites, which are our branches still. So this is the most common type of neuron. Our bipolar neurons, we still have one axon branch and then one dendrite with the cell body in between the two. And you don't need to know exactly where these are found or anything like that. And then unipolar have one process off of the cell body. So multipolar, many processes off. We only have two direct processes, and now we only have one direct process. So it kind of forms a T where we have our axons on one end. And these are really not technically dendrites. It's all kind of considered an axon. Okay, the actual impulse conduction. So we said that we can think of our nerves as in kind of our electrical system, and truly our nerve impulses mimic and are caused by an electrical current. So our membrane potential is when we have our ions on either side. In this case, we're going to discuss potassium and sodium. And so when we have an imbalance of potassium and sodium on the inside and outside of the cell, that creates a charge known as our membrane potential or resting potential. And then we talk about polarization, which means that there's an excess of positive ions on one side and an excess negative ions on the other side. So there's opposite charges. So in this case, rather than dealing with positive and negative, we're talking about two different positives, but they're imbalanced and their different charges still create a positive on the outside of the cell and a negative on the inside of the cell. So that's very important to remember, is at rest, the interior of our nerve is going to have a negative charge while the exterior is going to have a positive charge. There's also a high concentration of potassium inside and a high concentration of sodium outside. And so if you remember way back when we talked about cell transport, we talked about the sodium potassium pump. And so that's what's utilized by our nerves to maintain this resting potential. So our nerve isn't doing anything right now, it's just ready. So it's set up, it has the potential to react if it gets a stimulus. Okay, so once it receives a stimulus, we get channels opening up, and that sodium that's highly concentrated on the outside starts to rush in, which starts to turn the interior from negative to positive. And so as our cell membrane and the interior becomes more positive, we call that depolarization. So this is somewhat of a chain reaction. So if we get enough of a stimulus to cause depolarization, then kind of each channel will successively open. It's like dominoes. So positive enough to open the next one, positive enough to open the next one. So we get that kind of chain reaction, which is known as an action potential or the nerve impulse. Okay, so like I said, as that sodium is rushing in, it's causing our other sodium channels to open up. So more sodium is coming in and potassium is exiting out. So as the potassium exits out, those channels then shut to stop more sodium getting in, more potassium getting out. But kind of by the time that nerve impulse goes all the way to the end of our nerve, we've kind of flip-flopped. So now we're positive on the inside, we have all the sodium in there, and we're negative on the outside and have all the potassium out there. Sorry, guys. Okay. Like I said, yeah, we have a lot more potassium outside versus inside like the beginning, and a lot more sodium inside now versus outside like in the beginning. So because of that, we have to fix that imbalance and return back to our resting potential. So we use that sodium potassium pump again. And so we pump that sodium back out of the cell, potassium back into the cell. Our interior of the nerve becomes negative again. Exterior is now positive again, and we have our resting potential restored. Okay, this is kind of a nice to know, not a need to know. I'm not going to test you on this directly, but the term we're going to talk about is saltatory conduction. And so what we're talking about here is the way the myelin sheath works. So each sheath or each Schwann cell is connected by a node of Ranvier. And so the only place that ions and salt can pass through is that node. So when we look at an action potential or impulse, it kind of seems like it jumps from section to section. And so that's what saltatory conduction means. But that really is not a super important concept that I want you guys to spend a lot of time on. Okay, let's get into a few review questions. So the most numerous number of all glial cells are, and this is kind of a tough one because I don't think we actually discussed this quite yet. But the correct answer is B. And so astrocytes are super, super, super important. And so there's a ton of them in the brain to form that blood-brain barrier. All right, question 2. Which neurons detect sensations such as touch or heat and relay information about the stimuli to the central nervous system? Is it efferent neuron, interneuron, afferent neuron, or a Schwann cell? So it's going to be an afferent neuron. So remember, afferent arrives, so that's the one that's going to deliver the information. So our efferent neurons or motor neurons exit, so they relay messages from the brains to the muscles and other things. Our interneurons are only in the central nervous system, and there are a connection between our sensory and motor pathway. And then the Schwann cell is what forms the myelin sheath. So it's not a nerve ending itself, it just insulates our nerves. One more question for now. What is another term for nerve impulse? So repolarization, resting potential, depolarization, or an action potential? So the correct answer here is D, action potential. So repolarization is when we're using that sodium potassium pump to restore our resting potential. And so that resting potential is when we have our sodium on the outside, potassium on the inside. Inside of the neuron is negative, outside of the neuron is positive. And then depolarization is the kind of process of the membrane becoming more positive as the action potential or nerve impulse spreads down the cell. Okay, we're going to go through one more example talking about synapses before we go on to, or before I end this first section of our PowerPoint and we go on to our next section. So I'm going to flip back and forth between the next couple slides because this has a zoomed in version of our example, but I kind of wrote out our steps on the next slide. Okay, so step 1, the action potential actually gets all the way to the synaptic knob. So as it's traveling, we get constant kind of a train of depolarization one after the other. Eventually it gets to those synaptic knobs. That causes ion channels to open, and then calcium ions will enter our cell. So that action potential is spreading down the axon. It reaches our synaptic knob. Ions are released. Calcium comes in. Or sorry, ion channels open to allow the calcium to come in. So that infusion of calcium is then going to cause the vesicle to bind to our cell wall, and that will allow them to release their neurotransmitter into the synapse. So when we were doing our muscle chapter, we talked about the neurotransmitter acetylcholine, but there's multiple neurotransmitters that all have different signals that they send in the nervous system. So kind of starting again. So action potential, ion channels open, calcium comes in. That influx of calcium triggers our vesicles to go to the end of the synaptic knob, fuse with the membrane, and release their neurotransmitter into the synapse. Okay, like I said, there's different neurotransmitters within our nervous system. So we can have ones that are excitatory or continue our impulse, or we can have ones that are inhibitory or stop our impulse. So typically, if it's going to be an inhibitory signal, we're going to get potassium channels opening, which causes our impulse to stop. If it's excitatory, we'll get our sodium channels to open because that allows the membrane to depolarize. So that goes hand in hand with our depolarization, repolarization. So if potassium's coming back in, then our signal has stopped. If the sodium's still coming in, that means our signal is continuing. So then our receptor is going to release the neurotransmitter, just like we had an enzyme for acetylcholine. Kind of each neurotransmitter has its own unique enzyme that can clean it up, and then we wait for our next signal. So one more kind of quick run through. Signal kind of depolarization spreads down the axon, reaches the synaptic knob. Our ion channels open, calcium comes in. That influx of calcium causes vesicles to go release their neurotransmitter. The neurotransmitter binds here. If it's excitatory, we'll get more sodium coming in. If it was an inhibitory example, this would be replaced with potassium. And so basically kind of it binds long enough to send its signal. When it's done, it'll release it, and whatever's left will get eaten up by an enzyme. Okay, I think we are going to stop there for now, and we will pick back up talking a little bit more about our central nervous system.